Anand, is it? Oh, you just started recording, and now I'm going to make a wisecrack. um,
Yes, I don't
uh,
want to miss the introduction slide, so started the recording now.
okay, so yeah, I was just going to say, is it sort of like popcorn that you're supposed to, when you put popcorn in the microwave, you wait until the popping kernel sound goes down to a frequency of, one every 5 seconds or something, and then you say it's done. Is that what we're going to do with the participants jumping in? Because there's more people
That's what I was
joining
thinking.
every second now.
Yeah, that's what I was waiting to see once it starts tapering off a little bit.
Yeah, so the popcorn method.
Yeah, exactly.
Yeah. Well, welcome to everyone who's joined so far. We're just going to give it another minute or so because there's a lot of people still joining. See a lot of familiar faces. Welcome. Hope I live up to your expectations. If not, I'm safely behind my computer screen where you can't throw rotten tomatoes at me. I don't know if there's a rating system though afterwards, Anand, for, rate this performance.
I guess there's the reactions, I guess thumbs up at the end of the webinar.
Or maybe we can get it on IMDb
Yeah,
or Rotten Tomatoes. See what the critics say.
Okay, I think we should start now.
yeah, looks
Yep.
like popcorn is finished. Hello everyone. Thank you so much for attending this webinar on Beyond Confocal Microscopy. I'll start off by introducing myself. My name is Anand Matthews, and I'm part of the UHN Research Innovation course team. We work with the managers of our core facilities in a number of ways to ensure their long-term sustainability. This webinar is one where we're working to raise awareness of our facilities that provide access to instruments and expertise to help you reach your research goals. Next. UHN Research has several core facilities that are available for booking by UHN and community researchers. In addition to AMF and their vast selection of microscopes, we have over 20 cores that specialize in flow cytometry, preclinical in vivo imaging, genomics, and many more. You can check out our website for more details. Next. The Advanced Optical Microscopy Facility, or AOMF, is the largest core facility in its type in Canada. AOMF has over 40 instruments across 4 UHN sites, with 7 expert staff members and sees over 500 users per year. AOMF offers researchers access to a wide array of microscopy technologies and expertise. Next. James Jockman is a UHN research staff scientist and the manager of our AOMF facilities. James is passionate about teaching and training users with an emphasis on scientific rigor and reproducibility. In 2020, he pulled together a team of 4 other scientists from around the world to publish a Nature's Protocols tutorial paper on quantitative confocal microscopy, which is the subject of this webinar today. Please note that we are recording this webinar and will be posted on the Research and Innovation Core's website and on the AMF website. So I'm pleased to turn things over to James to begin this webinar. Thanks.
Okay, thanks. Let me see if I can share my screen. How's that? Do you see the right screen? See hand clapping from Beatrice, so should be okay.
Yes.
I'll just so thanks for that introduction, Anand, and thanks to Beatrice for all the planning of this webinar. I really appreciate it. Just one little correction, Anand. You were working off last year's notes. This talk is not about quantitative confocal microscopy. That was last year's webinar. Today, we're going to go beyond confocal microscopy. So if you're up for going beyond confocal microscopy, stay with us. As Anand said, we run a large core facility. We get hit with many different applications in a given year, and admittedly, you know, the confocal microscope is the tool for the job in many cases, but it's not the best at everything. So today I'm going to talk a bit about the disadvantages of confocal and how we can go faster, achieve higher resolution, more channels, go deeper, or maybe opt for something a little less expensive or less complicated than a confocal. Okay, and then last year, at almost exactly a year ago, February 28, we recorded this webinar, Guidance for Quantitative Confocal Microscopy, and you can go to our website and look that one up if you're interested, aomf.ca, and go to the resources section and you'll find it there. You'll also find a link to the Nature Protocols tutorial paper that Anand mentioned. So that'll give you a good grounding in all things confocal, but talks about a lot of related techniques as well. This is admittedly a very long paper. The five of us spent a long time working on this and trying to gather as much intel from our different perspectives around the world. So I hope that's a good resource for you. Okay, so just a quick review about confocal microscopy. So the confocal microscope, its main advantage over wide field microscopy is it has this very special property called optical sectioning. So this is a 12 to 15 micron thick sample, a kidney sample, and you can see this glomerulus has sharp features, but superimposed on that is blurry features from out of focus. So the confocal narrows in on a thin optical slice around maybe 1 micrometer thick. And as I focus through this sample, so this is a through focus series, you see when I go out of focus with wide field, it goes blurry. There's still lots of light there, but when I go out of focus on the end of the tissue with the confocal, it goes dark because the confocal is throwing away any out of focus light. And how does it work? You shine a laser beam into the sample and you generate fluorescence in the whole cone of illumination. So not just in the focus, but above and below as well. But you image that focal point through a pinhole onto the detector. So the pinhole is what throws away the out of focus light. So for example, light from the surface, it didn't come from the focus of the objective lens, so it's not collimated. So the pinhole lens won't focus it through the pinhole. So that's how the confocal gives you 1 pixel with only the in-focus light, and then you have to scan that pixel back and forth, or that focused laser beam back and forth, possibly change colors and build up a single slice, and then you can repeatedly change the focus and scan again to get a whole through focus series. You can do thicker samples like this, about 40 to 50 micron thick organoid. So you can collect these 3D volumes, and of course you can do 3D renderings and 3D quantification. Okay, so there are some great advantages to confocal. Obviously, you can do this optical sectioning. You have very good resolution. We're going to talk about that. It can be very sensitive, especially with new GASP or hybrid detectors. You can do some live cell imaging. It's great for colocalization because you're sure that the signal that you're colocalizing all came from the same focal plane. But there is this list of disadvantages that I want to work you through today and some of the alternatives to confocal. Okay, so first of all, it is somewhat slow. There's no denying that. And I want to talk about 2 aspects of speed. So if you're trying to capture fast dynamics like calcium sparks or intravital imaging of like a moving, breathing animal, or or any other fast dynamics, then you would benefit from a faster scanning system. But it's also relatively slow. So if you're trying to image very large fields of view, even for fixed samples, you may want to try to speed things up. So how slow is the laser scanning confocal? A rule of thumb is that when you have that focused laser beam, you probably have to dwell on each pixel for about 1 microsecond to get a good signal. And then a suggestion is that you should take at least a 1 megapixel image, 1024 by 1024 pixels. So 1 microsecond times a million pixels is about 1 second per image. So that's a single image slice. That doesn't actually sound that slow, but it can actually add up pretty quickly. First of all, to really get all the detail out of the image, out of a fixed slide, you may want to take more pixels, so that'll add 4 times more time, and then sequential imaging of 3 or even 4 colors. You might do some averaging to improve your signal to noise, and then you want to take a Z stack, and now you can get up to many minutes for a full 3D volume. And we do have people in the facility who are taking this kind of time to take really nice, beautiful, data-rich Z stacks. So that's great. Obviously, if you have dynamic processes you want to capture, that's not going to be okay. So one thing you can do is switch to a faster scanner. For example, a resonant scanning confocal. This will give you, so a resonant scanning confocal gives you about a 100 millisecond dwell time, 0.1 microsecond per pixel, not 100 milliseconds, 0.1 microsecond per pixel. And so if you were to compromise a bit and do just 512 by 512 pixels, then you can take an image in 0.03 seconds, so 30 milliseconds. So that's a lot faster. Now you're compromising quite a bit. You're doing a 512 by 512 image, and maybe you do simultaneous imaging of your 3 or 4 detector channels. And then you can generate 30 images a second, or maybe even 2 volumes per second, depending on how many Z slices you have. So here's an example. I don't know how well this is going to show up over the Zoom call, but here we have a zebrafish embryo with a beating heart. And you can see the dynamics of the heart here, but if you look carefully, I don't know if you're going to be able to see it, but there are red blood cells. So the blood cells are expressing RFP, and they're moving through these green GFP labeled blood vessels. And this is imaging 512 by 512 at 30 frames per second. You need that kind of speed to see those dynamics. This is taking a full Z stack, only 8 slices, but we were able to do 16 Z stacks per second by narrowing in on a thin strip of the image. It gets very busy. It's maybe a little bit hard to see what's going on in this 3D rendering. Definitely we're sacrificing quality here. It's a lot noisier. We had to do some post-processing to smooth it and so on. But you can capture those fast dynamics. The resonant scanner is not the only way to do this. You can also use some proprietary systems like the Zeiss ARIScan. We have an LSM 880 with an ARIScan fast mode, and that uses a multi-array detector, basically capturing multiple rows at the same time. And with their new 980, they can get up to 47 images a second, which is pretty amazing. Now that's only 1 color at a time, and you would have to multiplex between different colors. So if you wanted 3 or 4 colors, you'd have to divide this by 3 or 4. Still pretty fast. Or you could also use a spinning disc confocal. The spinning disc takes a laser beam. I don't have a lot of time to go through this in detail, but basically splits your laser beam up into about 1,000 little beams of light that are all hitting the sample in parallel and come back through a whole array of pinhole discs and then onto a camera. And as this disc spins, you sweep out across the entire sample. So spinning discs can also be very fast, but since they're camera based, again, unless you have a system with 3 or 4 cameras, you're limited to 1 channel at a time, or however many cameras you have. So this is just to point out that you can do live cell imaging on a confocal. You can do this very well on a resonant scanner or on the ARRI scan, or this was actually done on an older spinning disc confocal that we had. So hopefully you can see these microtubules kind of growing and shrinking and wiggling around, or this cell adhesion marker being recruited to the junction where these two cells touch. So you can do some nice dynamic imaging with these fast techniques. Okay, so here's just a couple slides from last year's webinar talking about optimizing confocal. When you have fixed samples, we encourage you to take your time and get nice high quality images. And so what we said at that time was that if you zoom in on a 512 by 512 image, you will see that it's actually very pixelated. It's not really very good quality, especially if you're going to try to crop into a smaller field of view. So what we said last year was the software will often guide you what the right number of pixels is. And for this objective lens and the field of view at zoom 1, to really get all the detail out of the image, you need 3000 by 3000 pixels, which is maybe going to take too long. So maybe we compromise a bit and do 2048 by 2048, make it less noisy by doing some averaging. Well, the first thing you should think about if you have fixed cells and you want to go faster is, this guidance was for giving you great quality, but microscopy is all about trade-offs. You don't have to go to a resonance scanner to speed this up. You can scrap this pixel suggestion, and you can say, well, I'm going to go down to just 1024 by 1024, 1 megapixel, or even lower. Our resonant scanner only does 512 by 512. So if you're still seeing the details you need to see, and that's what's going to give you the time to do a large tiling for a fixed sample, then that's up to you. You can scrap the line averaging, let it be a bit noisy, do some post-processing instead. You can try speeding up the scanner. You can turn on bidirectional scanning, which scans in both directions instead of just from left to right. So there are a lot of ways you can speed things up and approach resonant scanning speeds. You can also compromise on crosstalk. So what I said last year was imaging 4 times, once for each channel, is the cleanest way to make sure you separate everything. If you do everything simultaneously, turn on 4 detectors at the same time and 4 lasers at the same time, then you see especially the DAPI bleeds way into the green will bleed into the red, etc. But maybe you tolerate some crosstalk in order to speed things up, or maybe you compromise and do 2 colors together that are a little bit better separated and 2 other colors together. So you can speed up conventional imaging quite a lot. The resonance scanner is going to go even faster, but it'll be noisier, and you'll probably find yourself just adding back in some smoothing to make it look like what you're used to with your normal fixed sample imaging. So for fixed slides, I would actually say that if you don't dwell for 1 microsecond per pixel, if you go to a resonance scanner at 0.1 microseconds per pixel, you may not get the quality that you're looking for. And I strongly urge you to consider scrapping confocal entirely and physically sectioning your sample thinner so that it's appropriate for our whole slide scanners. So fluorescence whole slide scanning is very fast. You just drop off the slides and we digitize them for you within a few days. It's relatively high resolution, 1/4 micron per pixel, and we can do a large sample 2 by 2.5 centimeters, and we've digitized this entire thing at pretty amazing resolution, I'd say. So these are the individual nuclei of this tissue microarray. Now you do need special software to handle these kinds of large scans. So this is a 60,000 by 80,000 pixel image. And just to put that in perspective, that's at least 100 times bigger than the images you take on our smartphones these days. So try lining them all up beside each other, like line up 100 of your images off your phone and work with them all at the same time. It just doesn't work. So, but there are free tools for viewing, and then we have very powerful software called Halo for doing whole slide analysis, which you can also use, by the way, for big tiled and stitched images off a confocal, but not for a Z-stack. Okay, so you can tell the software what your what patterns you're looking for. In this example, tumor versus stroma, and Halo will unmix it all for you and then quantify just the regions that you want to quantify on a cell by cell basis if you want. So, okay, just to sum this up. Yes, the confocal is pretty slow. If you're looking for dynamics, you want to go to a resonance scanner, a spinning disk, or Airy scan, or there may be some other technologies related. If you're saying, well, it's slow because I want to do large fields of view on my tissue sections, I really think you should consider whole slide scanning instead of confocal tiling. I'll make more money off of you with you sitting there at the confocal for hours and hours, but it's probably not the best choice for you. I realize there's a lot of protocols out there, like for brains in neuroscience, where the protocols start with, okay, section it at 40 microns thick. That's too thick for our scanners. These are wide field scanners. If you don't have a three-dimensional question, then you can find ways of sectioning them thinner. You might want to take advantage of one of the histology cores that are available at our institute or elsewhere. Okay. So, oh, and I see a question. I usually don't notice the chat, but I see Anna's question. How thin does a section have to be for slide scanning? And we've had very good results up to, say, 12 or 15 microns, but definitely 40 microns is too thick. So standard histology is more like 4 or 5 microns, but it can be thicker than that. Yeah. Okay. So that's a bit about speed. And, we're also happy to work with you or go to your local core facility if you're from another institute, and the staff there can help you to optimize your scan settings to speed things up if you decide to stick with confocal tiling. Okay, I'm going to catch my breath and then go on to resolution. So a confocal microscope has pretty good resolution, 200 nanometers, and but we can improve on that. So what limits the resolution? The resolution is limited by diffraction of light. It's a basic physical property, and a rule of thumb is given by this Rayleigh criterion. The resolution depends on the wavelength of light and the numerical aperture, the NA of the lens, not its magnification actually. And I spoke about this last year as well. So the NA of this 20x lens is 0.8. If I were to, so this is a dry lens. If I were to plug in some numbers, like green light is about 500 nanometers, and this numerical aperture, this lens gives us 400 nanometers, and that's just a dry lens. And if I go to an oil lens, which has a 1.4 numerical aperture, and I plug those numbers in, that's where I can get down to 200 nanometers. So how can we go beyond that? And there are a number of super resolution techniques. I'm only going to talk about one, which is STED super resolution. It's probably the easiest to get started with, and still get a really like a very appreciable increase in resolution. Okay, so what is STED, or how does it work? So basically, remember I told you for confocal, you have a focused laser beam that gives you 1 pixel in the image, and then you have to scan it back and forth to generate the image. So the radius of that focus spot is your resolution. So what STED does is it superimposes a second beam at the emission wavelength of your fluorophore, but it's donut shaped, it's empty in the middle. So on this ring on the outside of your your imaging beam, where they overlap, the emission, this second laser light forces the excited molecules back down to the ground state before they have a chance to give off fluorescence. Okay, so it de-excites these excited molecules everywhere they overlap. And as you increase the power of your depletion laser, that overlapping laser, you squeeze your imaging beam down more and more, and you can get it down to something like 50 nanometers. So that's pretty amazing. All you have to do is kind of regular confocal imaging on our STED microscope with these additional lasers, and you turn on the STED lasers and you squeeze down your resolution. So these are some images off our system, confocal images of microtubules. Now I'm zoomed in to a high degree here, right? This is a tiny little portion of one cell. So there's a 5 micrometer scale bar, but you can, I think, appreciate even over the zoom with any quality issues you have. I think there's no denying you have a lot more resolution here. And maybe a little more meaningful for some people is imaging mitochondria. Again, focus into some mitochondria in one cell, like not even the entire cell. So we're zoomed in a lot here, and the outer membrane is labeled. But with confocal, it mostly looks filled in, whereas with STED, you can really see the outer membrane clearly. We have some people doing live cell STED as well on mitochondria. There's some particular probes for that. You can get a bit of a benefit in the depth direction as well. This is a side view. And here's an example of a punctate object, the nuclear pore complex with confocal. This is a single nucleus, part of a single nucleus. And then with STED, and then you can further improve it if you deconvolve it. We have Huygens deconvolution, and I hope you can tell that that's much cleaner, much smaller. There's a lot more detail in there than the original image. Okay, so yes, confocal is limited to about 200 nanometers, but there are techniques. I told you about STED. There are techniques for getting down to much better resolution, and you can further improve it with deconvolution. Be forewarned, though, that you do need some pretty rigorous sample prep. The first thing we're going to do is say, are you using the optimized cover slip thickness and some of the other things? So if you go back to last year's talk, we spoke about some of those sample prep optimizations. There's a question, is the mitochondria image using the STED in our facility? Yes, I took that image myself on our STED. Thanks for that question. Okay, so another downside of confocal and fluorescence microscopy in general is that we're typically limited to 4 fluorophores in one sample. So 4 channels. And the reason is that fluorescence spectra are quite broad. And as I mentioned earlier, if you image them sequentially one at a time, you can get very clean separation between 4 fluorophores, but it's very hard to fit in another one. So this Alexa 568 is orange. Some people say, well, can't I put something red in between? There's just not enough space to do it cleanly. And you get a ton of crosstalk if you try. Like you could also try to put a yellow one in, but you'll get a lot of crosstalk. So one thing you can do, and we are doing this, we're going to be getting a couple of new confocal microscopes with size 7 channels, so a near infrared channel. Older confocals, the detectors weren't sensitive above about 700 or 750 nanometers, so this would be useless. But newer detectors are sensitive to 800 or even further. And we've also upgraded a couple of our wide field scopes with a new lamp that has infrared emission and then a size 7 filter. So we can start adding size 7. We're also hoping to get a couple of slide scanners with size 7 channels. So okay, we've gone from 4 channels to 5. Helpful maybe, but why can't we do 6 or 7 or 8 or 12 channels like we hear about in flow cytometry? There they do something like compensation. And, the answer is, in principle, we can do more, but it's much more complicated. We have at least two spatial dimensions in addition to intensity, whereas flow is almost zero dimensional. It has only intensity. So we have maybe a 2D or a 3D image, maybe even 4D with time. So it's quite complex. And we also have a lot less light to work with, because in flow cytometry, you are adding up the intensity of a whole cell at a time, whereas we're looking at details within the cell, so spreading that out into different pixels in your image. So it can be done, but it's very slow and tedious and usually not worth it. But you can actually buy panels that are meant for like 7 dyes for tissue imaging. So these are similar to flow panels that you would get, but for tissue imaging specifically. Why, so maybe we just don't have the right microscope. But if you read the fine print, you do need multispectral imaging for these kinds of panels. And yet with most people want to scan them. They want to see the whole thing. They don't just want a little field of view here or there. So there's one scanner on the market. It used to be the Vectra from CRI and later PerkinElmer. Now Akoya owns this, and they've recently refreshed it. So I think it's probably going to be better than the old Vectra. They're now claiming in this 2.0 system, they've renamed it PhenoImager 2.0, and they can do 6-plex whole slide scans. The previous Vectra could only do small fields of view. They can do that in 12 minutes. That's pretty good. I haven't seen it yet, but that only gets you one more channel, right? I can already give you 5 on any standard microscope with size 7. So how do you take this to the next level? And there's a bunch of high multiplex imaging techniques out there. We have funding for getting one of these later this year. We're starting to demo them now. And these iterative fluorescence techniques, what they do is you label 3 colors plus DAPI, you image it on some kind of slide scanner, then you remove those 3 colors and you re-image it with 3 more colors. And there's a bunch of different instruments that have different methods of labeling and stripping the labels for the next round. They're commercialized by different companies. I'm sure I've missed a bunch. I know Brooker also has an instrument. There are some do-it-yourself techniques like IBEX. There's a very nice publication about IBEX where you can scan it on our scanners or use tiling on a confocal and do your own labeling. But I think this really becomes workable when you have the microfluidics and the microscope married together. So this is Akoya's implementation, and several of the companies do this, where you have microfluidics for all the labeling and stripping, and your sample stays on the scanner. So overnight, it would run through the entire sequence. And Akoya claims to do, you can buy this 36-plex panel for breast cancer, but they'll even go up to 100 fluorophores, 100 labels, not 100 fluorophores, 100 labels all in one sample with this iterative method. So I think that's maybe pushing your luck a little bit, but it's been demonstrated at least once. Certainly 30 is becoming more common. We also have an imaging mass cytometer, which instead of using fluorophores uses metal tags. And then it ablates a little piece of tissue and sucks it into a mass spec, which can separate out all these different antibodies. We have a world leader in this technique here in Toronto at Mount Sinai, Hartland Jackson, who's done some very nice work with this. You can look up this paper if you want. It's not my favorite technique because of how expensive the instrument is to maintain and to run, and but also just from the physics and imaging point of view, the resolution is limited by this. I'll go back to this, by this ablation size, which is about 1 micrometer in diameter, which gives you maybe 2 micrometers resolution, which is not great. And it's very slow, so you're limited to very small fields of view. I think these iterative fluorescence techniques will probably take over, but that's just my opinion. in the meantime, there is some nice work being done with IMC. It is complicated whether it's IMC or these high-plex fluorescence techniques. You have to start thinking about what you're going to do with this data. And inevitably, you end up doing sort of this cluster analysis with these t-SNE plots or heat maps. So you have to be thinking more along the lines of how genomics data is interpreted. But it is possible. So you can go from 4 fluorophores to 5, very easy with the size 7 channel. Then these high multiplex imaging systems can get you up to, I would say, easily 12 or 15 with very little effort. Maybe up to 30 on a commercial instrument or well above that. Okay, so that addresses the limited number of fluorophores. Let's see, how are we doing for time? Okay, so the next thing I want to talk about is the fact that we have a limited depth penetration. So this is disappointing to a lot of people. You have a sample and we say that confocal can throw away the out of focus light. So people want to do thick samples and image all the way through them. But we're really limited in confocal to about 100 micrometers, mainly because of scattering of light. So one thing you can do is go to 2-photon microscopy. Instead of single photon fluorescence, which is what's standard, this is basically regular fluorescence. So in regular fluorescence, you have a photon of light. It hits a molecule and excites it to a higher energy level. And then after losing a bit of energy, it then jumps back down to the ground state and gives off a photon. That's a bit longer wavelength. With 2-photon, you switch to a laser that's twice the wavelength. And now if 2 photons, each with half the energy needed to excite it, if they hit the same molecule at exactly the same time, then you can still excite fluorescence. And why is that helpful? So basically, the probability of those 2 photons interacting with the exact same molecule at the same time are really small outside of the focus. So they're also really small with a regular laser. You need to change to a femtosecond laser, pack all your energy into a very, very short pulse, very short time. And then you focus it down to a very small volume. And in that small volume, you have a reasonable chance that this will happen. And now you actually don't generate any fluorescence outside the focus. So we actually don't need the pinhole anymore. You can use a large area detector and collect all the light. You can probably achieve double the depth penetration. So these numbers depend very much on what tissue you're imaging into, but you can probably double your depth by going to 2-photon. Now, why does it work? It's mainly because you can capture scattered light. The thicker the sample, the more scattered light there is, and the scattered light will all be blocked by the pinhole because it doesn't look like it's coming from the focus anymore. Whereas the 2-photon, we know we only generated fluorescence at the focus, so let's just collect it all. Also, you're using infrared light at twice the wavelength, and that penetrates deeper into biological samples. This is still a great review paper from one of the OG groups, Watt Webb's lab, talking about multiphoton microscopy, almost more than 20 years ago, but it's still a great summary of 2-photon if you want to look that up. Our 2-photon systems are also great platforms for intravital imaging because you get that extra depth. But we've also configured our 2-photon microscope as an upright microscope specifically so that it can be used for animal imaging with these window chamber models. So the DaCosta lab on my floor here has done a lot of intravital imaging together with other collaborators and people from other labs. I see a question, can you get 200 micron penetration for collagen hydrogels? Maybe. It really depends on each sample, on their scattering and absorption properties. So it really depends how dense that collagen matrix is, but I think it's worth a shot. Okay, so let's say 200 microns doesn't impress you and you want to go much deeper. There are a series of tissue clearing techniques for fixed samples. Obviously, you can't use this on a live animal like I just showed you, but for fixed samples, you could start with a mouse brain. It's totally opaque. And after some steps, some clearing steps, you match the index of refraction very well in the end, and you can make it almost disappear. It's still there. It's just almost completely transparent. So Doug Richardson, I know him quite well. He's come and given a few talks for meetings I've organized. He's down at Harvard. He's got a fantastic review paper on this that I encourage you to look up. There are tons of techniques out there now for doing tissue clearing, some better than others. Even for samples that are not super thick, it can be beneficial. So this is a nice paper specifically on tissue clearing for organoids. And if anyone has tried imaging organoids, you'll notice that even 50 microns deep, it looks dark in the middle. And you wonder, oh, is there no fluorescence in there? It's there. It's just that you can't see it because of the scattering. So with a simple tissue clearing, relatively straightforward, you can actually image all the way down into fixed organoids as well. So that's something to look into. Now, when you have these large cleared samples, the organoids you can image on are confocals. You don't even need 2-photon for that. But for whole brains or for very thick samples, like 0.5 millimeter thick tissues, you probably need to switch to a light sheet microscope. It'll just be too slow to try to do this on a confocal. So a light sheet, the sort of original version of light sheet is that you shine light from the side, but you shine a sheet of light, like a thin, like a thin piece of paper of light across the sample, and then you image it from above. And now you're doing optical sectioning, not because of the optics of detection, but simply because you're illuminating only one slice of the image. So obviously you need quite a thick sample. You need a different geometry because you need to illuminate from the sides. But there are some versions of this, like this light sheet theta microscope, where you image at angles and then you have to do some reconstructions to see it. But that's what's described in this paper, the light sheet theta version. There are many versions of light sheet microscopy and they have very different applications. So for large tissue sections, like I was describing before, if people are doing Z-stack tiling of 40 micron tissues, well, why not make it a couple hundred microns thick, do a very simple tissue clearing, and then you can image it on something like this. We don't have this kind of system yet, but we do have funding for some kind of light sheet, and we're still evaluating which kind is going to be most appropriate for our samples. But for thick tissue sections, you can see very high resolution of these neurons across an entire brain. That would take forever to image at this quality on a confocal. But with light sheet, it's very possible. Now you're going to have huge data sets with light sheet typically. So before you consider doing this, you really have to make sure you actually have a 3D question. So it's common in neuroscience that you want to see connectivity. You might want to see vasculature, like vasculature connectivity. There may very well be 3D questions, but if you don't have a true 3D quantification in mind, like connectivity of relationships in 3D, then you shouldn't be doing thick sections. This is much harder than just sectioning it and doing whole slide scanning. So, and the data sets are really hard to work with because they're very big. So you can do it, but you really need a 3D question before you decide to do this. If you're going to try doing entire organs, I would say maybe start with thick sections and see how that goes, because that's much easier. Okay, so confocals have limited depth penetration, disappointingly only about 100 micrometers. 2-photon can basically double that. And that's great also for live imaging. But if you want to go even thicker, you can consider these tissue clearing and light sheet microscopy. Okay, and then the very last section is a real short one. Confocals, 2-photons, all these related techniques, these are pretty expensive and complicated, some of these instruments. So don't forget about just good old wide field microscopy, whether it's transmitted light or fluorescence. You can do very elegant experiments on a wide field. Here we didn't even label the sample, but we did very nice wound healing and showed in this review paper how if you quantify the area of the gap as a function of time with this very simple equation, you can actually get the real migration rate in microns per hour. No labeling at all. People often come to us and say, okay, what should I label my cells with in order to do this experiment? Maybe nothing, just good old phase contrast. And, you know, there are many applications of where wide field would be appropriate. So first of all, long-term time-lapse imaging is challenging on confocals and related microscopes, but a wide field does a very good job of that. It's much gentler on the illumination, less photo bleaching. I think it's also imperative that people review their slides on a wide field before going to confocal or slide scanning. You know, it's much faster and easier to pop these slides on a wide field. So if you don't have access to one in your lab, then we have several wide field scopes that you can sign up for, and they're around in your facility. You probably have them as well. And I would say you can even capture a fair bit of preliminary data on the wide fields. It's not that I don't want you to do confocal. We do. We're actually going to be installing several more confocals over the next year to keep up with demand. But just don't forget about good old wide field to start with. Okay, so I hope I told you how you can go faster, you can get higher resolution, more channels, deeper, and or maybe switch to just a less expensive and complicated system with these other techniques. So that's all I have. Thank you very much for your time. And I'm happy to take some questions. I have not been keeping up with the chat, so let me look if there's anything in the chat. And please feel free to add more messages in the chat. Let me see if there's any. Beatrice or Anand, if you're reviewing this as well, let me know if I'm missing a question here.
Yes, no questions so far.
Okay, I see one great presentation. Just wanted to clarify that using STED requires special probes and preparation. Regular prepared slides cannot be used by STED lasers. Please correct me if I'm wrong. Yeah, that's a really good point. So STED is relatively general purpose, but there's no point in trying STED if you haven't used the right cover slip thickness, the right mountant, but also you should consider particular fluorophores. You can't just come with, you know, regular live cells with GFP or, you know, surprisingly, the far red channel gives us the best results, like Alexa 647. And then Abirior, they're a competitor to our Leica STED microscope. Abirior sells special probes that are really optimized for STED, because not all fluorophores do this STED effect as well as others.
Yeah, I think there's two new questions that came in. First one is, when do you anticipate the new NIR confocal detectors to arrive?
I'm hoping easily before the summer. Let's say maybe by the end of May. I don't know. Remains to be seen, but we have a procurement in process for that.
Another question here is the resolution affected by the wavelength of the excitation or emission?
Yes, it is. Great question. So I think I very briefly showed resolution, the formula 1.22 lambda over 2 times NA. So lambda is the wavelength of light. That's definitely in the formula for resolution. On a laser scanning confocal, it's the excitation wavelength that matters. On a wide field or a spinning disk, it's the emission wavelength that matters in that formula. So your size 7 channel is going to have worse resolution than your Alexa 488, quite a bit different. But so you should plan for that. If you have fine details you want to see, put that in your lower wavelength channels. Yeah, good point.
There's another one also here is what is the resolution power with the high multiplex imaging?
Okay, so the resolution when you do high multiplex imaging. So the imaging mass cytometer in particular it ablates a spot that's 1 micrometer in diameter. So that's probably giving you about 2 micrometers resolution. When you think of a nucleus as being about 10 micrometers, you can see individual nuclei, but like the intracellular matrix around it or cell membranes, the resolution is not that great for it. But it's workable. It's sort of just barely good enough, I'd say. With these fluorescence iterative techniques, they're typically done with a high NA dry lens, like a 20x or 40x dry objective, which I did the calculation earlier. It gives you a 400 nanometer resolution. So similar to our slide scanner. Yep.
I believe that's all the questions in the
That's all,
chat.
yeah.
Yeah, but if anyone has any additional ones, please feel free to include in the chat, or otherwise, if you think about it afterwards, please feel free to email James Jonkin. I think you can see his email there on the slide there.
Well, you don't want them to email you, Anand,
I mean,
with
by
their
all means,
their microscopy questions?
everyone is also more than welcome to reach out to our email as well, the UHN Research Innovation Course team. We'll post in the chat as well if you have any
Yeah.
questions in terms of any AMF related activities or any other core research facilities you'd like to inquire about.
Great. Well, I want to thank Anand and Beatrice, both of you, for organizing this, and I want to thank everyone for joining me. I'm happy to have feedback or questions by email. Just give me a shout. Okay. Thanks, everybody.
Thank you for joining us all today, everyone.
Thanks, everyone.
Did you stop the recording?
Um,